I'm sorry, from the University of Edinburgh, designing a crawling cell using soft materials.
Okay. Hi. Hello. My name is Elsen. So I apologize for the biology content in my talk. And so here, this is the picture of a cell, eukaryotic cell. So you know that animals, plants are made of eukaryotic cell. The interesting thing in this cell is that these three cells, they all move. So they all can move. So the first one is this cell is called crawling. Is cell crawling. Let me play them. So what they did here, they put a cell on a solid, on a glass surface, and they observed that this cell can move. And then in the second type of cell motility I call it cell swimming. So this is a tumor cell inside a polymer, and this can swim. And as you can see, the swimming speed is typically much slower than crawling speed. And then the third type of cell motility is we call it grabbing. So here is a tumor cell inside the same polymeric network. Here the cell is able to form a finger-like protrusion to grab the polymer and pull the polymer to move. And so this is briefly the structure of the eukaryotic cell. So the important part of the eukaryotic cell is this thing which is called the actomyosin network. The actomyosin network consists of the actin filaments. So these are actin filaments, and they are all cross-linked by motor proteins in the middle. And then the second important point is these filaments are also polarized. So they are pointing in the same direction, which is away from the nucleus. So this is like a liquid crystals inside the cell. So this is the picture of a cell, and the arrows is just the average polarization of the actin filament. So in this case it's isotropic, and in this case so the cell is not moving, but in this case the cell is crawling to the right. And then so this is briefly the mechanism of cell crawling from the microscopic point of view. So here we have a cell, and the actin filament inside the cell are polarized to the right. So that means the cell is going to crawl to the right. And this is the actin filament, so it's pointing to the right. And the actin filament attached to this surface with focal adhesion. And then what happens is the minus end of the filament can depolymerize, and then the monomer goes to the front and polymerize at the front. So this makes the cell to move to the right. So this is the same picture again. So this is just to illustrate the importance of focal adhesion. So the focal adhesion basically prevent the filament from slipping as this monomer polymerize at the front. And then what happens here if you don't have focal adhesion, the filament can actually slip to the back. So as this polymerize, the filament slip to the back. So the crawling will not be very effective. And so the second, I show you there are three types of cell motility. The first one is the cell crawling, and this is the second one is cell swimming. So the difference in cell swimming is we don't have this focal adhesion, because the cell is inside a viscous environment, so they are not able to attach to any surface. So in this case, the actin polymerization is not very important for the motion of the cell. So this suggests that cell swimming might be driven by a different mechanism. So to summarize this, there are two active processes inside actomyosin network. So we call this type of liquid crystal an active liquid crystal because it's not in equilibrium. These two active processes force the liquid crystal to be out of equilibrium. So the first active process is the actin polymerization, which is important in cell crawling. This happens when the monomer at the back depolymerize and then polymerize at the front. So this creates a mass transport in the direction of the macroscopic polarization. And then the second active process is called the actin myosin contraction, which I will describe now. So this happens in this way. So imagine you have two actin filaments, and these actin filaments are cross-linked by the motor protein in the middle. And this motor pulls this filament to the right, and then pull this filament to the left. So basically, they pull this filament this way. As these filaments are being pulled, this also create a fluid flow like this. So this quadrupolar fluid flow is caused by the motor proteins pulling the filament. And then you can have the polymerization and then depolymerization at this end as usual. And then and then we can approximate the whole process as a forced dipole like this. So because if you look at this thing, so this is looks quite similar to the fluid flow generated by a forced dipole like this. So if you take average of these forced dipoles, you get an extra stress in the hydrodynamic equation. So this is the active stress. And now I will go through the model. So we use the hydrodynamic model, which has been developed with many people. So these are the variables. So C is the concentration of the actomyosin network, U is the fluid velocity, P is the polarization factor. So our model is quite simple. We just have a droplet of active liquid crystal. So you can imagine a droplet of this many forced dipole, and also because they are rod shaped, they have an orientational order. For simplicity, we introduce a free energy like this, free energy functional like this. So first we have phi of C, which is a double well potential similar to binary liquid. So if you see at this potential, they have two minima. The first is at C equals 0, which corresponds to the exterior of the droplet, and then the second minimum is at C equals C0, which is correspond to the interior of the droplet. And then here we have a surface tension term, and then here we have the usual Landau isotropic to polar transition. So if you see this term actually changes the sign as you go across the interface from here to here. So here the sign is positive, so it's isotropic, but inside the droplet this sign become negative, so this become polarized.
Why do you perform double for concentration?
Because I want to stabilize a droplet. So I want to confine the liquid crystal inside the droplet. So basically this two term is the binary liquid term which stabilize a droplet phase. And then the last term is we have the coupling between the polarization and the concentration. So basically beta here, if beta is positive, that means the polarization wants to point perpendicularly outwards at the interface. But if beta is negative, the polarization wants to point perpendicularly inwards. And then the equation of motion is, because C is a conserved variable, it obeys the continuity equation. So you have dc dt plus divergence of current. You have the convection current and then diffusion. And then the new thing is this active term, which is a current due to the polymerization. So this creates an additional mass transport in the direction of polarization. And then you have the usual equation for for polarization, which is similar to the Leslie-Erickson liquid crystal, and then you have the momentum balance equation. So rho du dt equal to pressure, and then the viscosity, and then you have two parts of the stress term here. The first one is the passive, the elastic stress from the liquid crystal deformation, and then the second part is the active stress from the actomyosin contraction. So now I show you a couple of simulations. So here we initialize a droplet on a non-slip surface. So here I make the droplet initially spherical, and then the polarization is initially uniform and pointing to the right. Because of the polymerization of the actin polymerization term, this causes the droplet to move to the right. And then what happens is if the polymerization rate is large enough, you can observe a thin layer of protrusion which is coming out of the droplet. So similar to the cell crawling that I show you. So this is just to plot the steady state. The steady state size of the droplet. So here you don't have protrusion and the droplet looks like this, and in this region you have a protrusion and the droplet looks like this. I also mentioned that we have a coupling between the polarization and the interface. Here I show you what happens if the coupling is very large. So if the coupling is very large, this wants to make the polarization to be perpendicular to the droplet, and this makes the polarization to be isotropic. And you can still see the thin layer of protrusion coming out of the droplet, but because it's isotropic, the droplet is not moving. So we have to reduce the value of beta to make it move. So this is the picture when you have so if you have a less coupling between the polarization and the interface, now it breaks the symmetry, and the droplet now can move. So this is just the the snapshot of the picture. And here, here I show you the effect of the activity, the actomyosin contraction. In the beginning, I told you that there are two active processes in the cell, the polymerization. The polymerization makes the cell to move in the direction of polarization, and then the second active process is the contraction, which goes into the stress term, and the role of the contraction can be interesting, like in this movie. So in this picture, the contraction can create an oscillating state which looks like this. Yes, I think I will stop here. So thank you very much.
Looks quite deterministic. Why no noise or?
Yeah, it's a deterministic mean field equation, so we don't have to put any noise.
You think it's irrelevant or?
Yeah,
It's even some noise. No, irrelevant.
yeah. Yes, I agree. So maybe maybe I think the best you can put the noise probably in the momentum equation. Like,
Absolutely, yeah.
yeah. I think
To have
that's
some
yeah.
random forces and to see.
Yeah, that will be better. Yes.
You know, the 3D things that you showed,
Yeah.
3D movies.
Yeah.
What essentially, and you know, it's a drop which is spreading. What sets the lateral scale? You know, you had a thing which.
Oh yeah.
What sets that lateral scale?
So you have the soft anchoring between the polarization and the interface. So it goes in the free energy. So the value of these parameters set the how strong the anchoring between the polarization and the interface. So, okay.
You said that you were using non-slip boundary condition,
Yeah.
and then you said the thing is moving and there's a profile. Can you say more about how you implement non-slip?
So yes. So it's a lattice-Boltzmann algorithm. So we use a lattice-Boltzmann simulation to simulate the fluid path. So the Navier-Stokes equation, we solve it using a lattice-Boltzmann equation. So we just I can tell
You can tell me later.
Yeah.
Yeah. Do you? I mean, you can observe these things, or at least some people can actually observe these things using some suitable probe, right?
Yes.
Right. And is there any intermediate theory between the Landau-Degen theory and being able to observe it? Can you do some kind of more microscopic studies
So
other than this Landau theory, or is this the first?
other people have done the microscopic. So I think the microscopic theory many people have.
All right, and when they do the microscopic theory, what do they do?
So basically normally they consider one active filament. So this thing polymerized, and with the polymerization rate K,
So,
then
I mean,
they measure the velocity of the cell.
no, but if you were trying to make a theory, I mean, how would you do an input to a theory that was on a lower level than a Landau theory?
Okay.
I mean, not a continuum theory, but some kind of, something that actually included the polymerization in some
Yeah.
sort of more explicit kind of way.
Yeah,
I mean, my underlying question here is to do with orders of magnitude,
I think
and whether or not you can actually put some orders of magnitude into your theory, which are in some sense plausible, and get some answers out that correspond to real numbers, and to know that there is some kind of connection between these two things, or whether or not simply you are putting in numbers in order to get out the numbers that you observe.
I think some people, right, have done the coarse graining process from microscopic to the hydrodynamic equation. So I think some group like Marchetti and Liverpool, they have done this connection.
Yes, you explained the treadmilling in terms of polymerization and depolymerization of the actin filament. What is the role of the actin myosin in this process?
The contraction. So actually the role for actin myosin contraction is more important for this second part of cell motility, swimming, which I didn't speak, which I didn't talk about, but in this motility, is driven mainly by the polymerization, but in this type of motility, it's driven mostly by
That is swimming.
contraction. Yeah.
So in the very last video that you had, it looked like you had the beta set to 0.
Yeah.
Did that mean that there wasn't any polymerization going on? It was only due to contraction, or?
Yeah, it's only due. Oh no, there is beta is the anchoring between the polarization and the interface.
Okay, but the polymerization is not affecting the motion then.
So I think in that movie, I still have the polymerization. That's why it's moving. But I also have contraction. Yeah.
No more questions? Thank you very much again.